Dear students, good morning. You will be fine at your respective place. Today, in continuation to our last topic, that is the molecular basis of this inheritance, we have discussed that about this introductory part, that is the genes. Now, today we are going to discuss about the nucleic acids and nucleotides. Now, these nucleic acids, अगर हम बात कर लेंगे, these nucleic acids, they are simply the polymers of nucleotides. They are the polymers of nucleotides. They are the polymers of nucleotides and are made up of these nucleic acids. They are the polymers of nucleotides and are made up of किस किस के बने हुए हैं? Number first, what we call as pentose sugar, which is a 5-carbon atom. Number second, nitrogen base. And number third, we have got phosphoric acid, या phosphate group. Now, these nucleic acids, they are the polymers of nucleotides and are made up of pentose sugar. They are made up of nitrogen base and phosphate group. And so we can say these nucleic acids, they are made up of these three different things. That is the pentose sugar, that is the nitrogen base, and the phosphate group. Now, let us try to see what is the structure of this pentose sugar. Yeah, this pentose sugar, अगर हम बात करेंगे, it has been divided into two parts. इसको हम दो हिस्सों में तकसीम कर सकते हैं. One is called as ribose sugar and another one is called a ribose, rather simply, and another one is called as deoxyribose. Now let us try to see the structure between these two. If this is the ribose and यहां से हम क्या बनाएंगे? deoxyribose. यहां से हम एक और बनाएंगे, that is this deoxyribose. See what we will see in the ribose sugar. This ribose sugar, it has got a 5-carbon compound. It has a what we call as a pentagonal shape. What we will see, oxygen, this is carbon, this is carbon, this is carbon, and this is one carbon. If we go on naming these carbon, this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and there is one more which actually is CH2OH, like this. Now this carbon, it has got one hydrogen and one OH group. This carbon, it has got one H and one OH group. This carbon again, it has got one H and one OH group, and this carbon 4 की अगर हम बात कर लेंगे, it has got only one H. So simply what we are going to do, we have to see the balance of this carbon. Now in ribose sugar, what will this is the structure like this. it has got these 5 carbon. This is what we call oxygen. यहाँ पर carbon first, if we go numbering, carbon first, carbon second, carbon third, and carbon fourth, and this is this fourth, and it's actually CH2OH. Now, in case of deoxyribose sugar, what we will see, deoxyribose sugar. Carbon, carbon, and see the difference. Where it lies the difference. Again, if this is the carbon 1, this is the carbon 2, this is the carbon 3, this is the carbon 4, this is actually CH2OH. And again, what we will see, it is H and OH. Here it is H, it is again H and OH, again it is simply H, and it is here H. So the difference between the two structures, in case of ribose, at carbon 2 we have got hydroxyl group, and in this deoxyribose what we have got, at second carbon we have got hydrogen group. This hydrogen groups, here we have got at second carbon, what we will see, we have got one hydrogen and one hydroxyl group, but here we will see the both, we will see they are both hydrogen atom. This is the main difference. अगर हम structure देखेंगे, this is the, these are the what we call as the pentose sugar जो हमने पहले बात की, 5 carbon sugar. These are 2 types. One is the ribose and another one is the deoxyribose. Now, this was the structure about this. Now, in case of second one, जो हमारे पास है, that is second one. That is the nitrogen base. नाइट्रोजन बेस हमारे पास जो है, दे आर ऑफ 2 types. Nitrogen base, we have got 2 types. First one, purines, and pyrimidines. So नाइट्रोजन बेस हमारे पास 2 किस्म के, एक one is purines and another one is pyrimidines. Purine जो हमारे पास है, there are 2. We have got 2 purines. There are 2. That is what we call as guanine. First one is the adenine, and second one is guanine. And pyrimidines we have got 3 total. There are 3 in number, there are 2 in number, and these are 3 in number. वो कौन से हैं? They are 3 in number: cytosine, thymine, and we have got uracil U. Now, when we see the structure, in case of DNA and RNA, there is the difference. In case of DNA, जो हम देखें DNA, RNA, there is the difference in these pyrimidines. DNA में हम क्या देखते हैं? We have got adenine, guanine, cytosine, and thymine. In case of RNA, we have got adenine, guanine, and we have got cytosine, and we have got uracil. So this is the difference between these nitrogen bases in the DNA and RNA. Now, that's the one difference. Second, जो हम बात कर लेंगे, also what we will see in this, what we call these purines, they are dicyclic compounds, rather dicyclic molecules, while as these are monocyclic molecules. purine जो है they are dicyclic molecules and that is there are the two rings or two cycles are found in these purines and there it is the only single cycle or single ring. That is why they are called as the monocyclic molecules. थर्ड बात अगर हम कर लेंगे, there are 4 nitrogen atoms in purines. There are 4 nitrogen atoms. अगर हम बात करें, there are 4 nitrogen atoms and they are positioned at 1, 3, 7 and 9. There are 4 nitrogen atoms. अगर हम बात, there are 4 nitrogen atoms. They are positioned at 1, 1st, 3rd, 7th and 9th. And here we have got only 2 nitrogen atoms and they are positioned at 1 and 3. This is third. Fourth, अगर हम बात कर लेंगे, now where these nitrogen bases जो है, where these nitrogens are added. चलो, we will draw again this sugar pentose. This is carbon first, this is carbon second, this is carbon third, this is carbon fourth, and we have got CH2OH. Now, these nitrogen, they are attached at this carbon first. This nitrogen base, they are attached at this nitrogen base. So, where we will see this carbon, this nitrogen atoms, they are what we call as, they are found at first carbon of pentose sugar, like this, at this place. They are at first carbon of pentose sugar. Pentose sugar, what we will see, pentose sugar, and ninth position of purine. लेकिन यहां पर हम क्या देते हैं, जो नाइट्रोजन एटम है, दे आर in case of purines we have got 9 nitrogen atoms. They are positioned at in purines at 1st, 3rd, 7th and 9th. But this nitrogen atom, it is at first carbon atom of pentose sugar and 9th position of purine, while as in pyrimidine it is also at first carbon of this pentose sugar. And जो हम बात कर लेंगे फर्स्ट position of pyrimidine. So in what we will see, there are 4 nitrogens, and in this purines they are positioned at 1st, 3rd and 7th purines, while as we have got 2 nitrogens and they are positioned at 1 and 3rd position of the pyrimidines. And this nitrogen is what we call it makes the bond with this first carbon of pentose sugar and 9th position of purine. Here it forms the first carbon of pentose sugar and first position of the pyrimidine. So this was second, that is this what we call as the nitrogen base. Now third जो हमारे पास है, that is the phosphate group. Third, that is the phosphate group, that is founder of, we can say, the phosphoric acid. Phosphoric acid. Chalo, draw the structure of this phosphoric acid. It is H3PO4. H3PO4. Draw the structure of this. This is P, this is hydroxyl group, this is hydroxyl group, this is hydroxyl group, and this is oxygen. So this is the structure of this phosphoric acid. Now what we will see कि ये कहां पर होता है, what we will see that one of the OH group in तीनों में से कोई one of the hydroxyl group, one of the hydroxyl, there are the three hydroxyl group, one of the hydroxyl group will make a bond with fifth carbon of the pentose sugar. For example, इस तरह हम देखेंगे when the structure comes into being. Carbon first, carbon second, carbon third, carbon fourth, and actually it is CH2OH. Now, one of these, what we call as hydroxyl group, they bond with this. They bond with this fifth carbon of the pentose sugar, and you will find the structure is, it becomes structured like this. Then it is P like this, OH and OH. So what we will see, how this phosphoric acid is attached in this pentose sugar. It is attached at the fifth carbon of the pentose sugar, and it is because of this that it is attached at the fifth carbon of this pentose sugar. Now, how these three things, that is this pentose sugar, that is this nitrogen base, and this phosphoric acid, are combined together to form a nucleotide. How they form a nucleotide, and these nucleic acids, rather they are the polymers of these nucleotides. Now, how they form, you will see. Now, how these three things, that is the pentose sugar, that is the nitrogen base, and this phosphoric acid, how they combine together to form a nucleotide. Now, let us see the structure of nucleotide. Pentose sugar, numbering 1, 2, 3, 4. What this is actually, it is actually CH2OH. Now, what we will see, that this nitrogen base, it is attached with this first carbon. This is the nitrogen base. And this phosphoric acid, what we will see, this phosphoric acid, it is attached at fifth carbon of this pentose sugar. P, OH, 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 and this is double bond O. Now, what happens? This H and OH, they combine together and they get eliminated in the form of water, and it gets eliminated. Now, the bond. Now, there are the two bonds, this and this fifth carbon and this phosphoric acid, and this first carbon and this nitrogen base. This bond which is found here, this bond, this bond is called as glycoside bond. This bond is called as glycoside bond. That is the bond with the carbon first and this nitrogen base. And this bond जो हम यहाँ पर देखते हैं, that is called as the bond which is formed there, that is called as phosphodiester bond. So here we will see the bond, this phosphodiester bond, it is the bond between the pentose sugar and the phosphoric acid, and this glycoside bond is between the pentose sugar and nitrogen base. Now what we will see, this is the nucleotide. Now what happens in the nucleoside? So nucleotide is this pentose sugar, this nitrogen base, and this phosphoric acid. Now, what is the structure of nucleoside? Nucleoside is simply nucleotide minus phosphoric acid. Minus phosphoric acid. So this nucleoside is without this nucleotide minus, this is minus, it is not bond, minus phosphoric acid. So या हम सिंपली ये कहेंगे it is, और हम सिंपली ये कहेंगे it is simply pentose sugar plus nitrogen base. नाइट्रोजन बेस. न्यूक्लियोसाइड में ये फॉस्फोरिक एसिड नहीं आता है, न्यूक्लियोटाइड में तीनों आते हैं, पेंटोज शुगर भी आता है, नाइट्रोजन बेस भी आता है, एंड दिस फॉस्फोरिक एसिड आता है. न्यूक्लियोसाइड में हम क्या देते हैं, सिंपली द न्यूक्लियोटाइड माइनस फॉस्फोरिक एसिड, और हम सिंपली ये कहेंगे पेंटोज शुगर प्लस नाइट्रोजन बेस. नाउ बाय नेमिंग, जब हम उसको नाम देना होगा किसी का, वी हैव टू बी वेरी मच केयरफुल, वी हैव टू बी वेरी मच केयरफुल व्हाट द नेम वी आर गोइंग टू डू. फॉर एग्जाम्पल हम यहां पर देखेंगे, वी विल टेक दिस एग्जाम्पल डीएनए का. नाउ व्हाट इज दिस, दिस इज डीएनए में हम क्या देखते हैं, इट इज डीऑक्सीराइबोस प्लस they have got the base that is the adenine base. Now this is nucleoside, it is a pentose sugar plus nitrogen base, it is nucleoside. Now we have got pentose sugar plus nitrogen base, and we have got this phosphoric acid, it is the nucleotide. It is the nucleotide. Now, what is this known as? This is called as deoxyadenosine. This is called as deoxyadenosine. Now, what is this called as? Deoxyadenosine monophosphate. Monophosphate. So by naming we have to be very much careful. दूसरी बात, what you have studied, this is the structure of the nucleotide, and this is the structure of the nucleus. Nucleotide is the what we call as the pentose sugar plus nitrogen base, and this phosphoric acid nitrogen base is attached as carbon first, while as this phosphoric acid it is attached to the carbon 5. But in case of the nucleoside, it is simply these two, that is this pentose sugar plus nitrogen base. Now again, one thing I want ये आपको सिखाने के लिए, why DNA is more stable than that of the RNA. If we draw the structure of both राइबो शुगर का भी और इसका भी, this is carbon first, carbon second, carbon third, carbon fourth, it is H and OH, it is H and OH, it is again H, OH, and it is simply H. This is the what we call as the ribose. Now in case of deoxyribose, where I will draw the diagram, the structure rather, this is carbon first, this is carbon second, this is carbon third, this is carbon fourth. Here we will see we have got hydrogen and hydroxyl group. Here we will see it is hydrogen and hydrogen. Now here we will see it is again hydrogen and hydroxyl group. Here it is simply the hydrogen, and this one, this is CH2OH. Now see the difference. यहाँ पर हम देखते हैं कि C2 और C3 में we have got two hydroxyl groups, and here we have got only one hydroxyl group. 2 में हम देखते हैं कोई हमारे पास फंक्शनल ग्रुप नहीं. ये कहा गया है, ये देखा गया है कि जितने नंबर ऑफ दीज फंक्शनल ग्रुप्स आर, उतना ज्यादा कंपाउंड जो होता है इट इज टू मच रिएक्टिव, वो अनस्टेबल समवट रहता है. कि जितना भी हम देखते हैं कि फंक्शनल ग्रुप ज्यादा होंगे उतना वो कंपाउंड जो हम देखते हैं कि दैट इट इज नॉट टू मच स्टेबल. जितने कम होंगे उतना हम देखते हैं कि उतना ज्यादा हम देखते हैं कि जो कंपाउंड जो होता है दैट is stable. Now in case of RNA, that is not too much stable because it has got many functional groups as compared to that of the DNA, because it has got less functional groups, so it is somewhat stable. So because हमारा जो अब ये भी hereditary character, that is the DNA जो है, that is somewhat what we call a stable. It is not too much, हम कहेंगे it is not these what we call as this reactive. It does not react, it is not too much reactive, but it is somewhat more stable as a compound. So this was about this nucleic acids and the nucleotides, and we will proceed further in this molecular basis. Today we have given you the description, or rather we say the structure of the nucleic acids and the nucleotides. So this was about this what we call as the structure of this nucleic acid and nucleotides. We will proceed further in the next video. Until then, goodbye. God bless you. Thanks.